Welcome to the last mini lecture of this week. We're wrapping up chapter 10. So in this section, we'll think about analysis of mRNA, thinking about gene expression. We'll work on in situ hybridization, showing where an mRNA is expressed. We'll look at different types of reporter genes that can track proteins in living cells. We'll think about mutants and then knocking down the expression with RNA interference. We can think about deleting or replacing with an altered version of a gene, so creating mutations, and doing that with a new system called CRISPR. We'll also think a little bit about transgenic plants, so genetically modified organisms, and how we can take advantage of genetic engineering to make some proteins in large amounts using that cloned DNA. So as we think about mRNA, we can get a snapshot of gene expression. So we can do RNA-seq or deep RNA sequencing. RNA is converted to cDNA, so we make the complementary DNA, and then sequence to make a transcriptome. And so this is just sequencing a cDNA library. We call it a transcriptome instead of a genome because it tells us what has been transcribed. And then we can do comparative analysis, comparing transcriptomes across cell types, developmental stages, disease versus health, or across species. We also have in situ hybridization. So in situ means in place. And so we don't just always want to know whether there's an mRNA present. Sometimes we want to know where it is in a cell or where it is on a chromosome. And other times we want to know which cell types it's in a tissue. And so it's pretty easy to gather a tissue. It can be harder to gather the separate cell types. So if you do a transcriptome of a tissue, you're going to get many different cell types in the mix. And this way, by looking in place, you can see which of the cell types that mRNA was in. So let's take a look. In situ hybridization uses complementary DNA or RNA with a label. And so we call it the antisense. So complementary, so we get that base pairing. So anti-parallel and complementary, and we see where the mRNA or DNA is in a cell. So here in the bottom left, we see marking nucleic acid sequences on DNA with different color dyes to see where different genes are on the chromosome. It's a fairly new technique to do it that way. We have other approaches as well. In the panel here in the middle with the green, we have labels showing where a particular mRNA is. And then on the right, we have an example that you'll look at in the capsaicin article. And so DRG is the dorsal root ganglion, and TG is the trigeminal ganglion. The dorsal root ganglion are the clusters of cell bodies along the spine that have the axons that go out and sense in the skin or other parts of the body. And so the dorsal root ganglion tends to go to the skin, so of the paws or hands, and there's sensation of capsaicin there. And then the trigeminal ganglia is kind of in the face, and that would also get things like taste buds, where you might taste the capsaicin. So they collected their mRNA to make a complementary library from the dorsal root ganglia, and that was where they found the VR1. The VR1 is the putative capsaicin receptor because it had some of the properties that they expected. But throughout the paper then, they are looking to confirm that the VR1 has all the properties they would expect of the capsaicin receptor. So here the pink is labeling complementary DNA by in situ hybridization to VR1. And what they knew is they expected the capsaicin receptor to be in the small cell bodies, which is one type of cell in the dorsal root, but not in the big cell bodies. If you look at the arrowhead, you see a light pink big circle. These are the big cell bodies with very little labeling for VR1. And then we see the smaller cell bodies with the dark label. So this was consistent with what they expected, that VR1 is expressed in situ or in the place that they expected. Similarly, they had VR1 expressed in the small cell bodies in the trigeminal ganglion. So we might also want to label where a protein is. not just which cell types, but where in a cell a protein is or which cell types. So these are alternatives to labeled antibodies. Antibodies that we have dye on can be really useful, but some proteins are really hard to get an antibody made to for various reasons. And so alternatives are always really useful. Here we can have a reporter gene, and it's often something that we add on to the sequence of a protein that makes it visible. A really common example is green fluorescent protein, or GFP. So we add this sequence, it's from a jellyfish that fluoresces. We add the sequence for that protein, and since all living organisms use the same codons, the code works in humans, although it's been altered to use the codons preferred by humans, so it gets translated more easily. And so the sequence for GFP can be added to a protein of interest, like we see here in the bottom, showing in green where all the tagged protein is. We also have different colors, so it's not just green, yellow, blue, and then if you add multiples of those, kind of like mixing finger paints or any paints, you can get a rainbow of colors. And a group generated mice that were transgenic, so they permanently had different FPs in the proteins in different cell types, and these made rainbow mice. So mice with neurons across the rainbow, which allowed tracing of these axons that you can see otherwise would be really hard to follow which came from which neuron. So to construct a reporter gene, you have your normal gene, some regulatory sequences, the regulatory sequences and your coding sequence. You would have you can replace the gene with a reporter, so you could replace something with GFP, or GFP is actually pretty small, so you could add it to the end of your protein itself. So you can either look for expression patterns by just replacing a gene with a fluorescent gene, or you can tag it as well. You can also make constructs that only have certain regulatory sequences and see which combination gets you expression in the pattern that you would like. So to do this, you're actually mutating genes or modifying them. And so we can use mutations, but we also have another class of mutations that are destroying the function of a protein. So it either can block the function or completely eliminate a protein. And so that can tell us if a protein was necessary for the function. And so an example of this, if you have a question of is this component necessary for a system to work, maybe think of a flashlight. Is it necessary to have batteries? What would you do to test that hypothesis? What if your hypothesis that it was necessary to have a bulb? So if you say it's necessary to have batteries, take out the batteries, see if your flashlight still works. If you want to say, is it necessary to have a bulb, you do a separate experiment, take out the bulb, leave the batteries in, and see if the flashlight works. In both cases, your flashlight would not work. And so you would say it's necessary to have bulb and batteries for your flashlight to work. We can do the same thing with proteins by taking out or mutating the protein. And if the system no longer works without that protein was necessary for the function. Deciding if something's necessary is a really important way to study biological systems. So this whole category of experiments, is it necessary, can be addressed with mutations. It can also be addressed with antagonists, so pharmacology, drugs that actually block the function. So mutations are not the only way to block function, but blocking function is an important way to decide if a protein is necessary to a system. So in classical genetics, chemicals were used to create many non-targeted mutants. So you'd have a bunch of bacteria or yeast in a vial, you'd add a chemical, and many mutations would occur. So you're adding a mutagen. And then you would screen and look for individual yeast or bacteria that lost the function, and then you would see where the mutation was for that organism, and that would tell you what was necessary for the function you were measuring. There's also targeted mutation techniques that we'll talk about next. So here we can target knocking down or reducing the expression of a protein with RNAi. We learned a little bit about dicer already, and so this is reverse genetics simply because it's the opposite direction of classical genetics. So now it targets specific genes. So RNAi is an example of this. You add double-stranded dsRNA to the cells. It gets processed and works with dicer to destroy the complementary mRNA and then reduce the production of that protein. So it's a knockdown instead of knocking out because your dicer may take a little bit to get rid of the mRNA, so a little bit of protein might get made. Usually you double check and do a control to make sure that your protein is knocked down significantly or gone entirely. Here's just an example of C. elegans. So the C. elegans has a property in the egg cells where the nuclei fuse. And this question was, is a particular protein necessary for that to happen? C. elegans eat bacteria. That's their normal food. And so if you have the double-stranded RNA in the bacteria, the worms eat it. In some cases, it gets incorporated into the embryos, and you can tell where that happened, and then see if it affects the function, and here that it prevented fusion. You can also delete or replace a gene with an altered version. So you can use regulatory or coding regions of genes to modify. You can modify them and insert them in an organism. If you do that, and it's a mouse, one strategy is transgenic mice using embryonic stem cells. So a transgene is a gene from another species added into an organism. And so if you transfect the embryonic stem cells, then those cells, they get fertilized and become the next generation. The whole next generation will have that transgene. This on the right is a mouse that aged prematurely by the transfected gene that they were experimenting with, compared to a normal mouse of the same age at the left that looks younger. So ES cells are the embryonic stem cells. They can be grown in culture. You have an altered version of the target gene. It can be taken up by some of the cells. If you take the embryonic stem cells and get more copies of them, isolate an early embryo and change the DNA by changing it with the transfected set genome, then you can make a mouse that's pseudopregnant, be a surrogate for those embryos, and then the mice at birth, some of them will have germline cells containing the altered gene. These might be embryonic stem cells that are not the zygote, and so it's at an early stage, but not all of the cells of the organism would have that transgene, and you can see that's represented by the patchiness of the orange. But if you mate these mice, some of them will have the transgene in their germline that will get passed on, and so you can make transgenic mice with all the cells having that transgene. But that's a lot of work. So transgenic mice work best in mice. Very few other species are we able to do that with, and it's very time consuming to do. So the RNAi and other techniques are very useful because that more experiments can be done with the same amount of time and money resources. So we can have conditional knockouts, so you might not just knock out a gene entirely, but have it triggered at a certain time and place. And so if you have a system where you change the regulation or you add regulatory elements, you can do very complicated experiments. We're not going to go into the details of this. We just don't have time to do every technique, but you could read more about it in your book. Very cool, but we just can't do everything. We have a new system that's kind of taking over that as well. The CRISPR system includes a Cas9 protein that uses a guide RNA. So this is reminiscent of other RNA and protein complexes, so the ribosome or the telomerase. where there's guided RNA that can be directed to a specific site. And now the Cas9 protein actually does a cleavage, and that cleavage can be used to swap in DNA. So a very useful enzyme, the Cas9 enzyme. And so this is much easier than classical transgenic animals. It can be targeted very specifically to the gene. And because of how Cas9 works, it swaps the gene into the original location. And that can be a problem with transgenic animals when the insertion is in a different place. So let's take a look at the top. The green blob is the Cas9 protein. We have a guide RNA, which we can do rationally because we can determine the base pairing much easier than you can determine how proteins will interact. And so using the guide RNA, you can both target where you want your DNA to go in and have the DNA that you want to insert. And so the Cas9 makes a double-stranded break. The altered version of the target gene that you've engineered in the lab, then by homologous recombination, due to the Cas9 activity, inserts the new gene. The other thing that you could do with CRISPR is have a recognition on your RNA that recognizes a regulatory region, and then you could have a protein where you fuse Cas9 to a transcription activator and turn a gene on, or fuse it to an inactivator to turn the gene off. So we can both target the regular Cas9 to different locations to insert genes that we target and very specifically modify, or we can modify the Cas9 and use the RNA to modify regulation. So CRISPR is a very useful tool. So it's unethical to use it in humans, so this has come up as an issue. We don't know what effects it might have across the world. It's been banned as a method for modifying human embryos. Science has a long history of eugenics and other approaches where people have been trying to make mutations that would be very unethical, but it has happened in the past, and so we're proceeding with a lot of caution, although even though China is part of the world organizations that have banned human embryos, one scientist there did use the technique to modify some human embryos, experimenting with preventing AIDS, which is a valuable thing, a goal, but unethical approach. And so sometimes the approach does not, the end goal does not justify the approach. And so that scientist has been censured severely by both the Chinese and world scientific communities. But we can still study genes that might be similar to human genes in a lab, in other models, in animal models. And so we might also think about ways to develop drugs. So FMR1 gene is a defective gene in Fragile X. Fragile X is a gene, is a disease where there is mental retardation because of mRNA not getting transported correctly to synapses. So neurons have a special problem with translation. If the protein is translated up at the cell body, it's hard to direct it to the right place, and it has a long way to go, and it slows down the response time. So some subset, maybe hundreds of mRNAs in neurons, are actually transported down the axon and at specific synapses where you can have local translation of proteins. This allows for extra regulatory mechanisms and speed. So in Fragile X, this FMR1 gene that helps move the mRNAs is defective, and studies of FMR1 with CRISPR in the lab, not on human embryos, but on cells that are expressing FMR1, have led to potential drug treatments. And so by looking at what happens in a model system in a dish with FMR1 and transport of mRNAs, potential drug treatments that would be ethical to try to use in humans with appropriate constraints can be developed. We also can cure blindness, which with transgenic plants, not transgenic humans, but in a wide part of the population where rice is the major part of the diet, rice lacks a lot of nutrients. But there's some people where that's the only food they can get, and it's difficult to develop an agriculture of other foods due to environmental conditions in those areas and other geopolitical conditions. But we can make a transgenic version of rice called golden rice that produces the precursor for vitamin A, which is necessary for making retinal, which is essential for vision. And so with this golden rice, if it could be taken up by the populations that need vitamin A, we could prevent blindness in many, many people. Other advantages of transgenic plants, you can add genes that would increase crop yields. That would allow us to feed more people with the same amount of cropland or leave more land wild. Some transgenes can reduce pesticide use, and then this idea of saving people from blindness with the golden rice. So transgenic plants have a gene from another organism. Some genetically modified plants or organisms, so GMOs, genetically modified organisms, some of these are not transgenic. I didn't stick the picture in, but there's a Simplot potato. There's a problem with potatoes, they produce acrylamide, and when we fry potatoes into French fries, it can become a carcinogen, which is bad for people. And the potatoes with the acrylamide also bruise more easily. This decreases the yield of food that humans will eat and decreases the amount that can be sold. And so these Simplot potatoes, they're just modified in a regulatory region to make less of a protein that they would normally make. And that reduces bruising, increases yields, and may reduce cancer risk. Although the amounts of acrylamide, honestly, are pretty small. And so the bigger benefit of these potatoes might be in the crop yields. An example, another reason plants are really nice model systems is that many of the leaves can be dedifferentiated so that they go back to being totipotent or pluripotent. They then can be put in environments to make shoots and roots, and so we have a whole plant growing from that. So if you make a engineered disc of plant or some cells, you can then make a whole plant from that. Much easier than a transgenic mouse. One last item, we have the other benefit of genetic engineering is that we can make hormones and other rare proteins in a lab. And so growth hormone is missing in some people that are not just short, but actually have medical conditions related to the absence of growth hormone. But it is hard to get. And so we had the only source for a while was from animals, and hormones are present in very low concentrations. And so getting enough growth hormone from animals required using a lot of animals to get the hormone. And it was, you can have reactions to cross animal treatments. Insulin, the same story. So people that have type 1 diabetes will die without insulin. Originally, we only could get insulin from animals, and we couldn't get very much at a time. And so it was very expensive. Now in a lab, we can genetically engineer organisms to make the insulin and purify it. And now we can have people's lives saved with insulin. We can also make antigens for vaccines. And now we can engineer RNA vaccines. So the Moderna and Pfizer vaccines are RNA vaccines. And so they're giving your arm or the injection of RNA that then makes the proteins in your body. And then those proteins are the antigens that your body then makes an immune response to. So RNA vaccines are something we're using right now to treat the pandemic. So to do this, we might have a protein. So we have a protein and we determine the amino acid sequence. We can find the sequence in the DNA database. We can make the right primers and clone it by PCR. We can then manipulate or introduce or alter that gene, insert it into a vector, into E. coli or other host cells and make a lot of that protein. We can do analysis on that, or then we could use it for treatments. So here's just another vector, another approach. So we have a vector with a promoter sequence, insert a coding sequence of interest, and then get that plasmid back in to some cells, overexpress the mRNA, and make a lot of that protein. So you could collect insulin, you could collect growth hormone. It gets to some ethical issues. Who should get growth hormone? It does treat disease, but it also could make some people taller. Would that be ethical? It's very expensive. If it's even ethical, it's going to be used only in a kid because the growth hormone doesn't have an effect after adulthood. Should the child decide? Should the parents decide? What if the parents don't agree? What if there's multiple parents? What if a grandparent doesn't agree? What if, who should pay for it? Should insurance pay for it? Should other people pay for it? So a lot of ethical questions about some treatments like growth hormone, and then other questions about who's going to pay, both for the research and for the treatments once we develop them. So we have a lot of tools, a lot of things we can do, but this raises questions about what we should do and how we're going to manage who's going to pay for things and who's going to benefit. So we've thought about exploring gene function, thinking about analysis of mRNAs with in situ hybridization, thinking about reporter genes, thinking about studying mutants and how that lets us explore what's necessary in a system or ask other questions. And then we can mutate and modify genes. We can do that with CRISPR much easier than we used to be able to. We can modify plants and also make other proteins. All of these raise really interesting questions about what we should do and how we're going to manage that going forward. That wraps up Chapter 10, thinking about isolating and cloning DNA molecules, DNA cloning by PCR, sequencing the DNA, and thinking about gene function. That also wraps up the end of mini lecture set 7 for February 22nd by finishing out Chapter 10 on analyzing genes.